Welcome to the first lecture on glycolysis and sugar metabolism here in biochemistry. So with this lecture, I will try to go through what is glycolysis and how do we get into the sugars that we want to do, and how do we get them actually to be, what should I say, turned from sugar into energy. So that's the main part of the next two lectures that we'll have here in biochemistry. So the learning objectives of the first lecture here in glycolysis and the following lectures. So first, we would like to see and make you understand how these metabolic networks they work in cells. Um, Maybe you will be able to do a little on modeling or at least understanding on the cake, which is this Japanese source of any kind of biochemical models. The biochemistry part is a bit of discussion of the glucose and why is it a good fuel source and so forth. And finally, to understand why is it important to understand the free energy that we talked about in the first lecture here in biochemistry. Why is it important to understand this during glycolysis? Because this determines whether it's exothermic or endothermic or exogonic or endogonic reactions. It also determines whether the reactions are more or less reversible. So that will get back to why this understanding of the standard Gibbs-free energy is very important. Now let's move on. So why at all talk about glycolysis? So glycolysis is a oxidation of glucose into pyruvate. And pyruvate, well, with pyruvate we can do a lot of things. And as you can see here in the figure, one of the things that is really important for pyruvate is that it's the key metabolite to take the chemical energy from sugars into something else. So in this situation, it could be lactate. Lactate you see very often when you are a little late for the bus. You feel it in your bones, or not in your bones, you feel it in your legs. And that gives you a good feeling to some extent because it shows you that you've been exercising, but the other hand, you really burn your muscles. So this is the way we'll talk about later how the fermentation in muscles take pyruvate into lactate and then recycle the energy. And this is the key part in this fermentation part. So this is one of the reasons we want to glycolysis. We want to have pyruvate. We also want to have ATP, and ATP is our, maybe remember, it's the key component in energy calculations. Now, how does it look? So when you need your sugar, then you see the top level here, and when you need your energy, you have the lower part. So if we start with the lower part, so we have glycogen, that is a polysaccharide that comprises of a lot of glucoses put together in 1,4-alpha linkages, and the glycogen is degraded into glucose. So here we get the glucose. And from the glucose, we go via the glycolysis into the pyruvate. So sometimes if you look at the upper part, we need to store our glucose. We don't need so much energy. And that could be when we're relaxing. So maybe the glycolysis have given us some pyruvate. The pyruvate can then be turned back to glucose via glycogenesis, we will hear about in the next lecture. And from the glucose that we see up here, we can then synthesize glycogen. We could also be that we needed the glucose. So that could be if we needed the glucose in the brain, for example, in humans. Then we could have like an efflux into our blood cells, and then we could transport the glucose around the body. So that's another way. So both of the things here are very important, and we would talk about influx and effluxes a little during this lecture, but we mainly focus on how do we oxidize glucose into pyruvate. Over on your right-hand side, you see the big picture that I like to show about biochemical metabolisms, and we're going to talk about the main highway in the middle. So it's also useful for biotechnology purposes. So in small organic acids, for example, in E. coli, Well, we will start with growing our E. coli on glucose or another sugar source. And as you can see, the glucose is then oxidized down to pyruvate that we have down here. And that pyruvate can then be fermented into lactate, we just talked about that, or alanine or other chemical molecules. And for example, it can be turned into 3-hydroxypropionate, so that we have here on your left-hand side. The 3-hydroxypropionate is often used to make biopolymers, and they can make now in E. coli 100 grams per liter in 48 hours. So 100 grams per liter, how much is that? Well, that's 10% of your bioreactor. Now, maybe you're impressed about that. Just think about that you can still make 30 to 40% of your bioreactor fuel of butanol in other reactions, not with E. coli, but with other organisms. So this is just an example of how the understanding of how glucose is oxidized into pyruvate can be utilized in biotechnology to see what we can understand more. So what we will see a lot about here in the glycolysis is protein complexes. So as you can see here, the A complex, it's a compilation of protein parts that has come together to form a protein complex. So this can be either individual enzymes, or it can be understand as the elements of a larger enzyme. What you see in the middle is a protein complex that we'll talk a lot about here. It's could be an example, could be the oxidative phosphorylation pathway, or the electron transport is also called. Now here you see a lot of proteins come together in a row, and you have to understand that maybe your molecule will come in here in the beginning, over here, and then it will travel through on the green part, green path, and it will come out at the end here where you have the arrow. So that could be an understanding on how, well, this situation, it's 5 different enzymes come together and form a protein complex as they're sitting in a row. The final part is that when you kind of like have scaffold proteins, so these are really important, and we're getting more and more understanding on scaffold proteins. And scaffold proteins helps other proteins to come together and form complexes. So there you can see if you have a mutagenesis in your scaffold protein, let's say that there was a mutagenesis here, fill it out like this, but then it wouldn't work anymore, and suddenly your whole protein complex would not work anymore. That could be very detrimental for your cells. So these are 3 types of protein complexes that we, to some extent, will talk about during the glycolysis, but definitely is super important to understand when we talk about biochemical pathways. So that means coming from one substrate to a product via several enzymes. And most of this is really the core of how we do protein engineering. Protein engineering is part of another course, so I'll not get too much into detail with that. But back, glycolysis. So glycolysis is taking monosaccharides that we have up here in the top part of your slide, mainly glucose, or we will turn the monosaccharides into glucose. Then the glucose will then be going on to 3-aldehyde 3-phosphate, and that part here we call the first stage. The second part, going from glyceraldehyde 3-phosphate to pyruvate, is the second stage of the glycolysis. Finally, in the next lectures, we will take pyruvate back to glucose as a glyconeogenesis, and following that, we will have the oxidation of pyruvate into acetyl-CoA, as you see here, and the further into the TCA cycle, which is also known as the citric acid cycle in your book. TCA, though, is an abbreviation, so it's a bit easier. The TCA cycle is the one that provides most of our organic acids, so like citrate, for example, as you see here, and also other organic acids that can be turned into amino acids and finally provide amino acids for protein building. The TCA cycles also then provides NADH and FADH that goes on into the oxidative phosphorylation that I just talked about, and that provides a lot of ATP. So the whole purpose of the next many lectures is to see how do we take glucose, as you see in the top up here, down to ATP that I just circled down here. So that's the main part of the glycolysis. The glycolysis is only the first two steps, so from glucose to pyruvate. Over on your right-hand side, you can see a more schematic figure on how these are collected in together, and it's more kind of like understanding that these are part of a network and they're not part of a individual pathway. It's a huge network of pathways that comes together. But before we dive into this, let's just talk about adenosine 5,3-phosphate, or also known as ATP. ATP is kind of like the currency of energy in biochemistry. It's the most important energy molecule in any living cells. So it doesn't only comes as ATP. So these molecules comes as the bases. So that could be arginine, guanine is the two most common ones. So that would be ATP and GTP. But it could also be the CTP and the TTPP. So all the nucleic acid bases are forming parts of these triphosphates. You can also see this on the nucleoside. The adenosine that you see here is a nucleoside that you also find in other pathways in the human, or in any cell actually, not only in the human body, but in any cell. And finally, we get onto the nucleotide. With the nucleotide over here is the AMP. AMP is the M for 1-phosphate. Now, building you see the phosphate group over here. So extending the phosphate group to 2 phosphate groups, that gives you the ADP and the D for the diphosphate. And finally, we get into the 3 phosphate groups down here on your right-hand side. And that gives you the ATP, which is a 3-phosphate molecule that we will talk a lot about, especially the conversion between ATP to ADP and the other way around is very important in understanding how does energy calculations goes on in the body, or the cells, I should say. It should be the cells, not only body. So ATP releases energy. So when you hydrolyzes ATP, so that means if you have here like a water molecule coming in and you hydrolyzes your ATP with water, then you get ATP to ADP. The ADP, that reaction releases 30 kilojoules per mole of ATP. So it also releases 3-phosphate, but that's not so important in this situation. The most important take-home message here is that it releases 30 kilojoules per mole. That means that the standard Gibbs free energy is minus 30 kilojoules per mole. It's something we can utilize later in biochemistry. So you imagine that you could use these 30 kilojoules to make other reactions. So that is the main and key point of ATP, is to use the 30 kilojoules per mole in other reactions in the cells. Now, where is the energy hidden in these molecules? So we have 3 types of energy hidden here. We have the resonance energy, we have electrostatic repulsion, and we have hydration. So the hydration is the one I just mentioned. So where you can see that if you go all the way to the lower right corner, you can see there's a little hydration of these pure phosphate molecules. Now, most of the energy is hidden in the resonance and the electrostatic repulsion. So if we take the ATP, which is on your left-hand side, and the lower one, because that's easier, if you look at that, you can see there are 2 negatively charged oxygens on the left-hand side molecules. So they are here and here. And then you can see in the middle, there is one here, and there's one here at the final on the right-hand side. So there's actually 4 negatively charged oxygen molecules. If you look through the different forms that are shown here on your left-hand side, you can see that these negatively charged are positioned at different oxygen molecules. So that means that we can take one negative charge. Let me just find a good one here. So if we take this one, put this in here as a double bond, That means that suddenly this one will be a lot of electrostatic, so we need to move this one here, and then we take that one out here as this electron here. That gives us this molecule you see up here on the just above here. Now, this of course can be done with all of them. So we could also do it here into this double bond, and then we could take this electron and put out here, right? And that gives us the molecule you see here. This change in this resonance structure and this electrostatic repulsion that will occur because this, if we take this molecule up here in the middle, there will be electrostatic repulsion between these two, which would actually push this this electron in here and then out here to give us a new molecule. And so these repulsions that are built into the whole structure, well, they are quite important to understand why it works like this. So just to repeat, I mean, this repulsion you see here, so these two negatively charged oxygen, they don't like to be together. So actually, this would rather go in here and then go back here, and then we get the molecule that we've shown down here. Now, why would this be more favorable? Well, if you look at the molecule here in below, that will be like the second from below. Well, the electrons are situated on both sides of the phosphate chain. So this is probably the, well, I couldn't say the most stable, but the most favorable, one of the most favorable situations to be in. But all this is changing up and down, and this is what gives us the lots of energy in these molecules. Of course, then when you cleave up with hydrolation, so if you cleave your molecule, let me just get another color. If you cleave here, for example, to give you this diphosphate, well, then you release some of this energy. And the same if you then cleave here, and then you get to this phosphate group, again, you release a lot of energy. The same, it's approximately the same release. There's a little more in the first release than there is in the second release, but it's not that much more. So just as a rule of thumb, it's 30 kilojoules per mole every time you cut a phosphate group. So one of the key factors is the cofactors. So we talked about cofactors before, and cofactors are super important in biochemistry. And here you can see an example why. So they are here stabilizing the ATP. As you can imagine, if you look into the magnesium molecule, has two positive charged ions. And actually, if you look on your right-hand side, you can see how this magnesium molecule suddenly actually interact with all three oxygen molecules. This stabilizes these molecules, of course, because then suddenly you don't have to do all this electrostatic repulsion and the resonance, because this is a stable molecule. This is one of the key factors where cofactors are super important to stabilize the double bond structure, and that enables maybe different kind of hydrolylations and enables the enzymes to work efficiently. Now, ATP is not the only molecule that have a lot of phosphate. We'll in the glycolysis also see another molecule, which is the 1,3-bisphosphate glycerate. So this can be cleaved, so you can have a hydrolation of your phosphate group up here on the top part of the molecule. So it should be here. So in this part of the molecule, so an hydrolation there provides the 3-phosphoglycerate, as you can see here on the right-hand side, and that reaction alone provides a staggering 50 kilojoules per mole of energy. And if you compare that with ATP to ADP, well, then maybe if we then take this reaction as we see here and couple that with the ADP to ATP reaction, so the opposite that we just discussed, that would require 30 kilojoules per mole. So we couple this reaction, so the ADP to the ATP, with the 1,3-bisphosphate glycerate hydroxylation to 3-phosphate glycerate, which gives you -50. Well, then the sum reaction of those two reactions coupled together is actually -20. So that means that the sum reaction of those two would be exergonic and thereby it could be running. This means that we can transfer a phosphate group here from the 1,3-bis-phosphate glycerate actually onto the ADP and into the ATP. Now, this enables us to do what is called substrate level phosphorylation. I just mentioned the word down here. There you go. And that's a very important reaction that we'll see in the glycolysis. So this gives us the possibility to build ATP from ADP in this situation. This is an overview of the phosphate-rich or the energy-rich molecules and how they relate to ATP. And if you want to do phosphate substrate level phosphorylation of ADP to ATP, you need to have a molecule with a lower delta G for hydrolysis. Now these are in kilojoules luckily, and the parentheses are in kilocalories if you rather prefer that. Now, as you can see, another molecule that we also talk about is the phosphoenol pyruvate, and hydrolysis of that from the -60 into the pyruvate will gives us the ability to do a substrate level phosphorylation of ADP to ATP. Again, it's a reaction we will see in the glycolysis. So with that, I conclude the first presentation here, and for the next presentations, we'll first do one where we talk about the first stage of glycolysis, and I'll go into see how we talk about the second stage of glycolysis. And especially in the second stage of glycolysis, we'll see one substrate level phosphorylation here in step 7 and another substrate level phosphorylation here in step 10. So where we have the 1,3-bis phosphate glycerate that I just talked about and the phosphoenol pyruvate molecule. So full oxidation of glucose into pyruvate will come in the next 2 videos.